So we think about phytoplankton community composition, there's a lot of work that goes into how we define it, but then also how we estimate it, but then also why. So I think it's important to maintain this question in the back of our minds. Why are we looking at the phytoplankton community composition? Because there's so many ways to go about it that letting the why drive our approach is crucial. And I wanted to include a few beautiful pictures. Maybe you've seen this or something like it. I think Kath had some in her talk, very similar, Gulf of Maine. And we get a little bit saturated, no pun intended, with these kind of flashy satellite ocean color pictures popping up on Instagram, NASA ocean color or whatever. But I mean, it's really amazing what we can see from space. And this is just a view of how the constituents in the water are changing spatially and temporally when we look at multiple days or whatnot that we can't see from the ocean. And intuitively, we know things about what the different colors and brightnesses in this picture mean, and theoretically also from what we know about optics and how it interacts with particles and dissolved matter, as you've all been learning. And how can we dig into that and be able to link what we see with what's actually there so that we can quantify what the different colors mean? And just a few more fun pictures, but from the ship level. So I'm sure you've all noticed the very green water here. Here's some green versus blue water. A few observed a few days apart off the coast of South Africa and on a little transect, so kind of moving out from the very strong productive upwelling waters to just towards the edge of the shelf there. And you can just see the amazing difference in the green versus blue water, the number of particles captured with an IFCB and the color too. And it's just nice to look at these side by side to be reminded that the ocean is so variable in color. And here's some blue and blue, slightly darker blue maybe. This is out in the middle of the South Atlantic Ocean. And there's kind of a similar number of particles, ROIs, cells obviously more on the right-hand side. But this was a small, well, not small, this was an eddy that we passed through, and it was almost entirely chaetosterous, the middle of the ocean, the relatively oligotrophic ocean. And there's some filtered water. You can see how brownish yellow it is. And Colin's not here, but when I worked with her years ago, we were out on the boat in Harpswell Sound, and I remember so clearly her looking at the water and saying something about what she thought might be in it based on the color and the brightness. And I was like, mind blown. Like, she's just looking at this green water and making, and she had some feeling for what was in it. So that sparked my interest too. Okay, what motivates our interest in PCC? I'm sure you've come across some of these aspects of oceanography when you're thinking about PCC. And I'll come to how we define it in a minute, but really referring to the size and the taxonomic composition of the phytoplankton, which both influence a variety of factors in the ecosystem on all different scales. So variability in particle carbon flux, so particles sinking, different size particles obviously sinking differently or aggregating differently and then sinking. Food web dynamics, different phytoplankton are supporting higher trophic levels differently. And then this on the right, this is a sort of an overview of topics that could potentially be addressed using hyperspectral remote sensing in a review by Dearson et al. And I just added blue stars to highlight where phytoplankton community composition plays a role. So how is it defined? Many ways. You might hear phytoplankton size classes, PSCs. Also, PSC stands for photosynthetic carotenoids, a lot of P's and C's and S's. Phytoplankton groups has been used in some publications. Phytoplankton functional types has gone a little bit out of style. We tend to not use that as the umbrella term for which phytoplankton are in the water because it might imply biogeochemical function. So it's one way to describe phytoplankton, but not the only way. And you'll notice in papers that these terms might change based on the user. Obviously, the bottom line being that we want to define phytoplankton differently or in more detail than just the total biomass. But there's a lot of ways to do that, and that's fine. Just to be aware that there's variability in the literature on the definition. And lately, and in work with colleagues on a review paper related to this topic, we're using phytoplankton community composition as the umbrella term that can include sizes, taxonomic groups, etc. Feel free to adopt PCC as well, but no pressure. How is it measured? You heard a nice talk from Sasha Kramer in week 1, detailing all these different approaches: microscopy, pigments, cytometry, imaging flow cytometry, optical signatures, molecular methods. So I'm not going to step through these in detail, but of course can refer to her slides. And then what about units? Why would we want to define something by absolute concentration or carbon? And why would we want to define something by a relative, a fraction of the total? And when you see a paper that's reporting on phytoplankton community composition distributions from space, what are the units? What can they tell you? How do the results and the units, excuse me, inform us and push us towards the questions that we're trying to answer? Sometimes you might see probability of occurrence. Maybe that's useful for harmful algal species. A dominant group, what can that tell us? And in some cases, consistency in the unit is really what matters. So if we're trying to look at change through time, then we need to compare apples to apples. So how can we scale up all these ways that we measure phytoplankton community composition in situ to regional and global views? And one answer is optics and remote sensing, as you know. So we can use optics as a tool to expand our spatial and temporal resolution, although I included an asterisk because spatial coverage is greatly enhanced by satellite remote sensing, but of course spatial resolution is finer with an in situ measurement, which presents also a set of challenges when we're thinking about comparing discrete samples in satellite remote sensing. And this is a nice figure from one of Patrick's papers showing different ways that we can make in situ measurements on different spatial and temporal scales. And then this kind of picture of what those different spatial resolutions look like. And it really comes into play strongly when we think about using satellite remote sensing data. And, in general, we sort of group together satellite remote sensing to think about studying the ocean. But when we really narrow in on certain coastal regions, does it start to become less appropriate to use a satellite pixel at a very near shore environment, for example? And how can we merge these different measurement types? This is also a figure that I like that is adapted from a figure Mary Jane Perry presented years ago in the optics class. And optical oceanography is sort of a niche field of oceanography. You may encounter other oceanographers, physical oceanographers, biological, and you say you're an optical oceanographer and they don't really know what that means. But I think this is a nice way to visualize that it's really this tool that connects what we can measure to what we want to know. So it's heavy on the development of algorithms, trying to understand optical proxies, and how can we make these connections between what we measure and what we want to know. There was only a paper airplane aircraft, so that was the best I could do for airplane clip art. Okay, so there's all our motivation and background for PCC. Hopefully you're on the PCC train. There's been a variety of work over the past 10, 15 years working towards assessing phytoplankton community composition from space. I guess 20 years is from, yeah. And most previously published methods fall into two main categories, so abundance-based and spectral-based, where abundance-based is estimating PCC via relationships between the phytoplankton communities and chlorophyll, and then chlorophyll is in turn estimated from reflectance. And then spectral-based is linking some feature or some using the spectra in some way to directly assess phytoplankton community composition, either from RRS PCC or via IOPs. Of course, measuring in situ, you could measure IOPs directly and look at PCC, but from space, we're starting with reflectance. So I'm not going to go through comprehensively all previous studies, but I have some tables and references in here so you can have a picture of previously published papers for your records. But I'll just talk about a couple examples of how this has been done. So for abundance-based, this is first PSCs, so phytoplankton size classes. I think they've probably come up in the last few weeks, but the pico, nano, microplankton. So the smallest up to 2 microns, and then 2 to 20. Some debate about around the best cutoff for nanoplankton, and then cells larger than 20 microns considered microplankton. So in these in this figure on the left, you can see the chlorophyll concentration on the x-axis, and this is from Bruin, Bob Bruin et al. 2010, and the fraction of pico, nano, and microplankton, also pico and nano combined, relative to chlorophyll A concentration. Sorry, I add the pico and micro. Generally, we have more chlorophyll concentration, and the fraction of microplankton increases. So I will correct that before I post the slides. And then from these relationships, you can then create maps of the 3 size classes from a satellite view of the 3 size classes, or the fraction contribution to chlorophyll. And in this case, and in other papers that have similarly defined pico, nano, and microplankton, they're defined using accessory pigments based on a couple of papers, 2001 and 2006, that attributed certain phytoplankton accessory pigments to certain taxonomic groups, and those taxonomic groups to certain size classes. And there are several assumptions that are included in defining size classes by pigments. And we'll keep thinking about that. So we did some work to evaluate how effective it is to define the size classes using pigments. And to do that, we compared, or we independently assessed contribution to size classes using cytometry data. And 2 things to notice. These relationships, when we use cytometry data rather than pigments, are a little bit less, for this data set anyway, are more variable. And so that is indicating to us that using pigments and assigning them to size classes is might have limitations in how accurate that is. And then also on the bottom, this is just not the fraction, but the total chlorophyll concentration from cytometry of these different size classes relative to total chlorophyll, or compared to total chlorophyll A. And to note that, you know, everything is increasing when there's more stuff. So something to keep in mind. And then we're interested maybe in the fractions of different groups across different chlorophyll concentrations. An example of abundance-based PCC estimates from space, now getting at taxonomic groups. So this is a similar approach now, where the authors have compared phytoplankton community composition as defined by pigments to total chlorophyll A. And they're also, this is SST, but they also use sea surface temperature in this paper. So they looked at both relationships to temperature and chlorophyll and incorporated both of those relationships with each taxonomic group into their algorithm. And then you end up with figures like this that can show distributions of the taxonomic groups on a global scale based on satellite sea surface temperature and satellite chlorophyll. They also did a really nice uncertainty assessment in this paper, which is important in this type of work, propagate uncertainties. But what is really limiting here at some point is the fact that the phytoplankton communities are defined by pigments, and we'll come to the sort of pros and cons of that in a little bit, and why that becomes a bottleneck at some point. So spectral based PSCs, some work by Arya Chiatti, who you all met, yes, and then together with Anik Rico also, and there's also a paper from 2006 they did together. So they devised a spectrally based size parameter to estimate the contribution of large and small cells to the total absorption, where their size factor, SF, varies between, well, 0 and 1 and higher and lower also, but it's like a relative contribution to the total of small and large. And when it's high, there's more small cells, higher contribution of the absorption signal of picoplankton. And so this was an approach to extract information on the size classes directly from the spectra. There's also was some work done in 2005 and 2008 by Alvain et al., where they looked at anomalies in RRS signal. So the RRS, multispectral RRS from satellite relative to chlorophyll, so that they could look at the differences in shape in the RRS signal, independent from the concentration of total biomass. And again, they used pigments to define the phytoplankton groups and associated anomalies in the RS with different pigment-based phytoplankton communities, and then could go and estimate them from satellite. And then, so you end up with a map like this, and maybe some parts of this map make sense to us based on what we know from in situ measurements, and in other ways maybe not. But they're also defining phytoplankton or their results by looking at the dominant group. So then we can think about what are the pros and cons of estimating the dominant phytoplankton group. What's that useful for? What is it not? What does it not tell us? And then some recent work by Lange et al. 2020, looking at using a PCA approach with RRS data and using in situ cytometry-based measurements of different picoplankton taxonomic groups. And they devised this algorithm and had really nice matchups for their in situ and also for satellite data to be able to estimate the concentrations of Prochlorococcus and Echinococcal and autotrophic pico eukaryotes, which they then applied to satellite RRS. This is a review by Mao et al. In 2017. Kind of, it's now becoming out of date because there's been work done since then, but it still has some nice information, overview information on phytoplankton community composition, phytoplankton groups, as they refer to approaches from satellite. And I just wanted to point out that the development inputs for all of these algorithms are not the actual PCC properties that we're estimating from satellite. So at some point, we have to think carefully and really consider the fact that we're trying to estimate something from space that we're not measuring in situ and building an algorithm, and there are proxies along the way. And in some cases, it's well, often it's totally necessary and that's why it's done, but also is a consideration when we think about moving towards more accurate algorithms, because we can tune the method itself and the statistics, but if it's apples and oranges when we're comparing our outputs to our algorithm inputs, that will be a threshold or a limitation, fundamental algorithm limitation. I won't go through this, but it's just for the slides so that you have it. This is from a review paper that is in prep by Ivana, who you also met, looking at all the different ways that the community estimates phytoplankton community composition, both from remote sensing, which is majority multispectral, because that's mostly what we have. That's mostly the type of remote sensing data that exists. And then in situ hyperspectral methods that perhaps could be applied to hyperspectral remote sensing data when that becomes more and more ubiquitous. Okay, now we're gonna think deeply about the advantages and limitations of defining PCC using pigments while you have a chocolate, so that you can get away. Manual, the first time I ever went manual to the conference and gave a lecture and we did a hands-on experiment. That's the only thing that I ever remember to talk through. Sasha touched on this in her lecture, and I think I was skimming through some of the other talks and I saw someone else, maybe Ivana, had also said, please be thoughtful or something along that line with your use of phytoplankton pigments. And so I'm just reiterating it in my own way here, that there are advantages and limitations of defining PCC with pigments. And there are some real advantages. We have pigment data from all over the world, and it's easy to standardize the method to extract HPLC pigments. And the absorption by different colored pigments is directly related to why the water is a different color. So it's very important. But pigments are not equivalent to cellular carbon or biomass. The relationship varies with taxonomy and with physiology. So how the cells have adapted to their environment. They may be making more pigments when there's low light so they can collect more photons. And so then we measure more pigments, but it doesn't actually mean that there's more of that type of phytoplankton. And the taxonomic resolution is limited. So there are some relationships or some pigments that are found in certain groups, but they're not unique. And although we do have, there exists on CBAS and other repositories a lot of pigment data from all over the world, we still are collecting discrete samples, a liter of water as once a day or whatnot. That has to be filtered, stored, processed in a lab. And it's actually much lower sampling resolution than like a continuously operated optical instrument. Then this just sort of to get us thinking about moving on to the next part of the talk. And these are some gaps identified in assessing PCC from ocean color from space in a review by Brecker et al. In 2017. And some of these gaps we're working towards. So missing capabilities. We have PACE launching in January. I'm sure Jeremy mentioned that. That will be hyperspectral. Traceability of uncertainties. I hope that uncertainties have been part of the conversation here for the last 4 weeks, but yeah, I encourage you to, we'll come back to that later in the talk also, but I encourage you to always keep uncertainties in the back of your mind. And then I don't know why I'm working backwards, but the gap one information mismatch. So that's what I was just referring to with the algorithm inputs versus the algorithm outputs. How can we think about addressing that? And that's also changing quickly as we have more and more types of data that are easy to collect on large scales. So I just wanted to talk a little bit specifically about hyperspectral data because it's all the rage, but that's not really new. So there's a paper almost 20 years ago, The New Age of Hyperspectral Oceanography, Chang et al. In Oceanography, pointing out that, hey, there are things that we can see in the water, or to do with the water that we cannot identify using multispectral data. And so this is an example. These hyperspectral curves were created with Hydrolite, which I think you all learned. Yeah, Hydrolite software, originally created by Kurt Mobley. And in this example, this is like a shallow water example, but showing that very different water with different bottom types looks the same to a CWIFS or similar multispectral sensor. But in a hyperspectral sensor, maybe there's a feature that we could actually identify. There's the same review I referenced earlier in the talk, but the Dearson et al. Review in 2021 is looking at living up to the hype of hyperspectral. And they go through all the different ways that hyperspectral data are treated and all the different approaches that have been used, and some that perhaps could be applied to remote sensing. And this, again, is just a table for your reference. So these are approaches to extract information from hyperspectral data, not specific to remote sensing, but some that could potentially be applied to remote sensing data. And yeah, just to go through in a little bit more detail, some of the approaches that are used with hyperspectral, both reflectance and IOPs. So Vandermeulen et al. 2020, and then Dearson et al. Soon after did some work to look at the apparent visible wavelength of RRS. And this is an approach that uses all the wavelengths of hyperspectral data to classify different spectra. So it's like an integration under the curve to find the apparent visible wavelength of each spectra, but where the value of the wavelength is not important. And then Dearson et al. Sort of used that AVW approach to come up with this quipped quantitative metric for quality control of RRS. And they have a big library where they can use the spectral shape of RRS to assess whether a given spectra is realistic. And then if there's a spectra that is measured, say by a satellite, but not necessarily, can be applied in situ also, it can be flagged and then analyzed further or just removed from the data. There's something that's not working. I think QUIP is actually incorporated into HyperOC, the software that Niels has worked on. It is, yeah. There's a group at NASA led by Nima Pahlavan, and they do quite a lot of work on inland and coastal waters, looking at how to apply hyperspectral measurements to assess water quality metrics. So their focus is mostly on chlorophyll, phycocyanin, and then also estimating IOPs. So not so much into different taxonomic groups, but some with the cyanobacteria blooms in lakes and inland coastal waters. And they're taking advantage of the information in hyperspectral data by using a neural network that called a type of neural network called a mixture density network, where a distribution of values is produced by the network and then combined to create phytoplankton absorption at different wavelengths. And they found that this approach reduces bias and eliminates a problem of invalid data points as compared to quasi-analytical and GIOP inversion methods. And so some of this is being kind of folded in and used in the upcoming efforts to assess hyperspectral or to use hyperspectral measurements from space. And then as I'm sure you've been discussing, phytoplankton pigments are what drive spectral absorption features in many cases. I'm sort of open ocean biased currently, so I'm sorry for people who are thinking a lot about detritus and non-algal particles and CDOM, but in the open ocean, most of the particles are plankton and there's not much CDOM. And so the shape of the absorption and thus the reflectance is really driven by the phytoplankton composition. And so here's a plot from some data from Vittigeri 1990 showing the spectral shapes of different major accessory pigments. And in this case, the photosynthetic carotenoids and photoprotective carotenoids, PSC and PPC, are grouped together. So fuchusanthin and peridinin are grouped together in the purple curve, and PPC would be like zeaxanthin and photoprotective pigments. And then the PE is the little orange one with little black curves, and it's like phycobilly protein from like cyanobacteria. So we see this and we think, okay, there's absorption that's differential absorption at different wavelengths, and in particular, in the red. So in absorption, absorption in the red, you can really see that chlorophyll C and B and A have individual absorption peaks. But if we're trying to invert for the presence of all these pigments, does this problem become ill-posed? So we have these groups, PSC, PPC, okay? Then we start talking about all the individual accessory pigments. There's a lot more curves now. And this is some work led by Kale et al. 2020, showing that the degrees of freedom in an inversion for these data is heavily determined by the error associated with the data that you're using in your inversion. So the work that I did, bunch of the work that I did during grad school was to build off this method from Hopner and Sathyandranath. They have some papers in 1991, 92, and 93, where they did a Gaussian decomposition of absorption measured in the lab, first with cultures, so like a benchtop spectrophotometer, and then using samples, and this is the 93 paper from the ocean, Gulf of Maine, slope waters. And they assigned the center peak locations of these Gaussian functions to wavelengths that are the known local maxima absorption of the different chlorophylls. And they found that it worked quite well. So we kind of adapted this method to the ACS using particulate absorption. One adaptation was to add a curve for non-algal particles or non-pigmented particles, because the ACS particulate absorption spectra include all particles, whereas the benchtop spectrophotometer, you can get at just the phytoplankton absorption. So we included the NAP signal, and we also did some other work to account for some smoothing that happens within the ACS. We wanted to undo the smoothing so that we could really be sure to extract information on these small, subtle shoulders and local maxima that represent absorption by different pigments. So in this method, we estimate chlorophyll A, chlorophyll C, and chlorophyll B, also grouped photosynthetic, the grouped carotenoids. And here's an application. This is showing chlorophyll A, but we have similar maps for the other accessory pigments. Chlorophyll A from absorption from the AC, and many others did a ton of work for collecting all this data with an ACS. And there's also other NASA funded expeditions in this map. And I should say that Gaussian decomposition isn't the only way that we estimate chlorophyll from ACS. I'm maybe even working on that, but there's also the line height approach that compares similarly. But in addition to chlorophyll A, using the Gaussian decomposition, we can estimate chlorophyll B and C and look not only at the absolute concentrations, but also at the intensive variables. So the ratios of different pigments, either set chlorophyll B to C here, but chlorophyll C to A, B to A, etc., to look at the relative contribution of different pigments where we've collected data with the ACS. And maybe that tells us something about the phytoplankton community. And so as we know, phytoplankton pigments are attributed to different groups. And here's just a table showing how maybe chlorophyll B and C could be useful to help us separate or to help us identify the presence of different phytoplankton taxonomic groups. So although dinoflagellates contain B and C, different types can, otherwise, the different major taxonomic groups of phytoplankton are separated by their presence or by whether or not they contain chlorophyll B or C. So what's important to note here is that chlorophyll B and C are not a useful tool to define the phytoplankton community in situ, but they might be a useful tool to inform us, like as an input, piece of input information to an algorithm about what's in the water or not in the water. So if we have tons of chlorophyll B, relatively, then it's not it does not indicate a lot of diatoms and vice versa. And then we extended that to include the Gaussian functions during a inversion of RS. So this is, I think you did an inversion lab. This is starting with RS, and this is an approach using the coefficients from Gordon et al. In 1988. So from RS to small RS just below the surface. And then from that, we quadratic equation, we get the U, which is equal to backscattering over absorption plus backscattering, whereas the RS is approximately equal. But this equation accounts for the other factors, that the F and Q factor that would be here in the RS equals B back over A plus B back. And then U is broken down into all its components, and where we would have an eigenvector or a basis vector for phytoplankton absorption, then we include all the Gaussian functions. So this is obviously only possible with hyperspectral RRS, because now we're including a lot more unknowns and we need more wavelengths and degrees of freedom and information. And then using a cost function, we can find the relative contribution of the different heights of all the Gaussians that gives us our measured RRS, and then back out the absorption of the different pigments. One challenge is that we don't have a lot of information in the red wavelengths from RS because the water absorbs all the red light. So now we're using the blue and green wavelengths, and the absorption by different chlorophylls and pigments in the blue and green is a little more muddled. So just adding to the fun challenge of the whole thing. And then these are the data that we used in this version of the algorithm development from different locations around the world. You can decide whether or not that's a global algorithm, but here's an example of relative chlorophyll C to A estimated from this set of variable hyperspectral RS measured in situ with a hyperpro. But most pigments, pretty much with the exception of, I don't know, zeaxanthin, divinyl chlorophyll B, which is in Prochlorococcus, are correlated with chlorophyll A. So chlorophyll A goes up, all the pigments go up. So maybe we can just create a relationship between chlorophyll A and each individual accessory pigment, draw a line through it, and then we can estimate all the accessory pigments directly from chlorophyll A. We tested the Gaussian method against that, and there are some publications that have done a similar thing to look at accessory pigments on a global scale, and it can be useful. But what's also interesting is that if we can estimate the accessory pigments independently from chlorophyll A, then we can go past this information that we can get from total biomass and the fact that everything correlates with it, to see when the community in the water is, with pigments at least, are different from what we expect based on these average global relationships across pigments. So to that end, there were some notes, some nice points highlighted in the Kale et al. Paper related to this effort of going beyond chlorophyll A and how to get there, and how the error or uncertainty in the data play a major role. And careful data collection is obviously important, but at the very least, careful quantification and propagation of error during algorithm development is needed. Error is the difference between having 4 to 5 degrees of freedom rather than more than 60. And there's also a recent follow-on paper from just a few months ago, looking at degrees of freedom in RRS, not just absorption spectra. And unsurprisingly, there are only a few, because there are only a few in absorption spectra also. So, related to this idea that everything in the ocean is correlated, the interest is to go beyond that correlation, beyond chlorophyll, and see how the conditions in the water deviate from that first order axis where everything is correlated. So my take-home messages from hyperspectral measurements are, the question is not as simple as how much information can we extract, because in some cases, wavelengths, well, in all cases, wavelengths are correlated with each other, and we may only have limited degrees of freedom in a given spectra. But are there approaches that we can take advantage of where the added information in hyperspectral measurements are relevant to a given research question? Are we looking for an interesting feature in the green, or something that indicates the presence of our seagrass in the area that we care about? Or are we doing a kind of quip analysis to see if the spectra are reasonable? Or are we just going to extract 1 or 2 pieces of information from the hyperspectral data, but maybe with a reduced uncertainty relative to multispectral, etc.? All right, everybody alive? Breathing? Stretching? Okay, power through, we're almost done. All right, so I just wanted to touch on like a few, I guess what I would call new methods, although there are some things that are just not new, but newly popular, let's say, in the world of PCC from space, and share a little bit about what I've been doing. But first, just a couple of interesting things that have popped up. This is a recent paper, and it's sort of a new version of ChemTax, and they made some significant improvements. First, they coded up both version 1 of ChemTax and version 2, 1.95 in R, so it's fully open source. And if you haven't heard of ChemTax, it's a method to assess phytoplankton taxonomic groups using pigments. So maybe a powerful tool to take advantage of all the pigment data that exists to define phytoplankton groups. People have mixed feelings about ChemTax because it requires seeding the algorithm with assigned ratios between pigments and the groups that are in the water. So people have mixed feelings about it, but this paper really addressed some of those challenges nicely, and they use a simulated annealing, which is rather than just a steepest descent, like minimization algorithm. In simulated annealing, the minimization randomly chooses a new point in the algorithm space and tests all different locations within that space, so that it can find the minimum without being caught in the global minimum without being caught in a local minimum. And they tried with a kind of basic data set and then a more complicated one, and they had good results. So anyway, maybe it's useful if you're a ChemTax user or thinking about using a lot of pigment data. Molecular methods, I just included these for completeness. These are from Sasha's lecture, so I won't go through in detail, but of course, molecular methods are becoming more and more common in thinking about big global open ocean work. as the approaches become more ubiquitous and more things sequenced, cheaper technologies, etc. And she also pointed out that how well the molecular methods are linked to the phytoplankton community may be dependent on your location. So like everything, there are caveats. And then we come to my favorite, but try not to be too biased, which is the use of imaging systems to, well, this is all cytometry, but the use of imaging flow cytometry to assess cells. And this is a nice review from a few years ago by Lombard et al., where they're looking at all the different techniques, not just imaging, but to quantify in situ, actually counting and observing individual cells to quantify planktonic communities. And they're all different instruments now that are becoming more and more common that can capture different regions of the full phytoplankton size spectrum, and here all the way up to the big stuff using the UVP. And yeah, not all are imaging, but they're all quantitative in some way. But the IFCB, which as we know is here, kind of looking at the medium and large phytoplankton cells, 8 to 150 microns. So nanoplankton and microplankton, excluding the smallest nanoplankton. And I know you looked at some data from Collins IFCB. Yes, I was admiring those. This is the data that I've been working with quite a bit, collected by Emmanuel and the group at UMaine. And it's powerful data because we're looking at actually what's in the water. And of course, there are caveats like anything and uncertainties in how well we're quantifying numbers of cells that we're seeing and the volume or biomass of each cell and its taxonomic identity. But it's in a way a more direct measurement of the cells in the water. But of course, we have all the big data, and this comes to machine learning. And I saw some look through the slides. Patrick gave a really nice talk on machine learning. Well, I hope it was nice. The slides look nice. I'm sure it was great. And so, one branch of machine learning that we use for these images is convolutional neural networks, which are well suited to imagery data. So this is what I've been working on quite a bit the last couple of years. Not this review paper, but this is a nice review paper that Lee Kartbos and others are on, looking at different, or just assessing machine learning for plankton images. And there have been also a variety of other papers recently that have reported on efforts to use deep learning, machine learning to assess the imagery. I'm gradually adding things to this GitHub organization, which is called IFCB Utopia, where Utopia stands for User-Friendly Tools for Oceanic Plankton Image Analysis. And there's currently up there, there's a repo called CNN Demo, and you can go in and play with it. It's just a Jupyter notebook, and there's a toy data set, which is 5 types of phytoplankton, 1,000 images of each, so it's really lightweight. You can play on your own computer with it. And it walks you through how to use the CNN to look at the images. Once we get into millions of images, then I use cloud computing, or we use cloud computing. Patrick has a bunch of experience with that, which is sort of another can of worms, but also interesting. Happy to answer follow-up questions if people have them about that later. And then as a result, we end up with millions of images that are classified, and for now we're focused on diatoms with the imagery. And with that, we can look at these relationships that in the past we've been in the community using pigments to define diatoms, and now we're using the imagery. So both methods have uncertainties, but here what I want you to notice is that as we look across multiple orders of magnitude of chlorophyll concentration, diatom carbon generally is increasing. So more chlorophyll, more diatom carbon, but there's huge variability. And for a given, bin of chlorophyll concentration or a given value, there's multiple order of magnitudes of variability in diatom carbon. So, you know, with a pigment-based method where we don't see this variability because it's by definition, the diatoms are defined relative, you know, by the pigments. So this is new information that's extremely valuable because now we can try to get at, well, why would there be more diatoms when the chlorophyll is 1, concentration of 1 milligram per cubic meter? You know, what is there a condition or a place or a time where we have 10 milligrams per cubic meter of diatom carbon versus 0.3? Hint, ancillary data is important. And also not just diatom carbon, but we can look at other proxies. So this is the imagery it's used to create the blue dots, but then optical proxies for POC and C phyto as a function of chlorophyll A. And this is helpful as sort of a sanity check, like, well, we shouldn't have more diatoms than total carbon phytoplankton carbon, and we shouldn't have more total phytoplankton carbon than total particulate carbon. So interesting. Also, imagery and non-imaging cytometry can be, these types of data can be merged together. A nice paper from Niels last year using all the data from the North Atlantic to merge together cytometry and imaging flow cytometry to capture a full phytoplankton size distribution type data. And, you know, these types of techniques are becoming more popular, and they're creating large data sets that can be used for in situ validation of remote sensing estimates of phytoplankton size distribution. So moving beyond the pigment-based definitions of pico, micro, and nanoplankton to this using sort of real measurements of different size classes or size distributions, maybe more realistically, of phytoplankton and total particles. And also as a part of this paper, there's a really nice comparison of all the different ways that we estimate size using optical techniques. So here's the Ciotti and Bricot size factor that I mentioned earlier, chlorophyll A, gamma from the slope of CP, PSD slope, mean size. Yeah. Yeah, volume distribution probably highlighting the presence of different communities that are present together and where your cell size is falling out into bins that represent kind of the different taxonomic populations, or maybe even size classes based on dividing or something. So recently I've been using machine learning in 2 different ways. One is to train convolutional neural networks and apply them to lots and lots of IFCB data. And the other is to then use those classified images, in particular diatoms, and use machine learning to be able to estimate or predict the presence of diatoms using input data such as environmental and optical ocean color information. So different types of machine learning, and I'm learning as I go, but I have found that in fact that deep neural networks are not always the way to go. They certainly are for the imagery data, but when we only have several thousand data records rather than hundreds of thousands or more, when we're comparing diatoms to their environment and their optical properties of the water that they're in, actually sort of tree-based or gradient boosted methods are more appropriate for fewer data points and tabular data where the inputs are related to the outputs in a way that we know something about. And this also raises the question of how then do we compare all of our data types? How do we even mash them all together? Everything we've measured in situ to build this type of machine learning relationship. We have continuous ACS measurements we get pigments from. We have TSG running all the time on a ship that we get temperature and salinity from. We have the IFCB running all the time, collecting information about the community, but every 20 minutes, and then maybe we have to merge samples because there's not much in the water, etc. And then we're going to link it to satellite, or we're going to include model input or reanalysis input, or data as an input. This is just one approach. There's different ways to do it, but I'm using this, what I call a long track gridding method. Yeah, it looks very beautiful, and it's an approach to have a regular interval for all data types that can be put together. So all data that are both the inputs and the targets to a machine learning algorithm can be referenced to a grid point, a long track grid point that's unique in location, but also in time. And then we also have this very challenging question, thinking about phytoplankton community composition from space. We want global algorithms. We want to, read what PACE is going to send down to us and just produce phytoplankton community maps across the globe. But how well do our training data represent the globe? And is there something we can do about it if they don't represent the globe perfectly, which of course they don't. So in this example, the background is the Modus Aqua 2020 annual mean of chlorophyll concentration. And the black dots are all the places where I have IFCB and optical environmental input data. Whoops. And then on the histogram is showing in the background, the gray chlorophyll concentrations from the Modus satellite data. And then the green is the chlorophyll from our long track ACS that's used in the algorithm development, and they don't match. So one approach that I'm working on is a weighting, which works with tree-based machine learning, where you can manipulate your input data to represent the distribution of your, in this case, of the whole globe. Stay tuned to see if it helps, works. Yeah. Mm. Right, like the one I showed earlier with the different RS, and how those don't actually capture all the, yeah. Yeah, right. Well, and not just the shape, but then the combinations of all the things. So when the temperature's high, but the salinity is low and the shape is this, did we ever measure that in situ? No, but that's what the satellite sensors on multiple satellite platforms tell us. So, yeah, it's a good point. And is there a way then that we should, and we will mask our algorithm outputs to say this algorithm is being applied in a place where the conditions were never present in our training data. So use with caution, or don't use, or whatever. Is there a way that we can come up with an uncertainty for that, or is it just trash? And thinking carefully about that. There's been quite a bit of talk about that related to PACE going up, and how we need to be thoughtful about presenting algorithms that produce products that people will use. And obviously, if they're not accurate, it's not useful. This is just kind of another view of the same challenge. So here's a 2 dimensions, TS, no 3, there's TS and chlorophyll of all of the data from the map I showed earlier. And then the black points are a subset of all of the data in the TS diagram, where we also have IFCB data. And so there's there's major sort of oceanic regions that are missing from the data that we have. And this also is sort of tied into the approach that some people are thinking a lot about, which is like ocean biomes and, you know, Longhurst, but also new versions of kind of a Longhurst type approach, bioscapes, and should we be treating different regions of the ocean differently when we're thinking about algorithm development? And there's a push-pull there, because if we put all the data together, we have better dynamic range, we have more points, but does that always make sense? Or should we have an oligotrophic version, or should we have a, you know, whatever? So different ways to think about that. Probably eventually, when we have more and more and more data, it will make sense to parse out regions of the ocean when we're developing algorithms, because you'll just never find this certain combination in some place, but we'll lose dynamic range too. So, and I've been obviously alluding to it and talking about it, but the inclusion of ancillary environmental data. So it came up earlier at the end of Kat's talk during the questions, but inclusion of temperature, salinity, mixed layer depth, mixed layer depth DT, water mass history, light, KD, I don't know. You could brainstorm all the different things that you could maybe estimate from space or from model reanalysis and incorporate them into a model. And we did that for this region in the North Atlantic using just chlorophyll, temperature, and salinity, which we could estimate from space currently, but there will be more products coming in the future, and there are other products now, to make a map of diatom carbon that's not just a function of chlorophyll A, but that is a function of temperature, salinity, chlorophyll, using this machine learning approach, a shallow neural network, and there are some different features. It's sort of a proof of concept, but it's the idea. Maybe we'll see things pop up that that don't show up when we just estimate diatoms from the first order correlation with chlorophyll. And can we convince ourselves that they're real, and can we quantify the uncertainties? So we have to quantify the uncertainties. We've tried to do this by propagating all the error in our target data, in our input data, and in the machine learning algorithm, or whatever algorithm you have itself. In this case, this is a median relative error of 65%. Is it good enough? What are you doing with this diatom carbon estimate and its associated uncertainty? Is that meaningful with that uncertainty or not? And thinking beyond like an average or median value, you know, per pixel error, and that's becoming in the conversation. That's more in the conversation and is planned for PACE. You know, can we provide errors that are specific to a pixel and a location? This is just an AGU opinion piece that is physical oceanography heavy, but they had some nice points that I think probably to all of you sound like old news, but for some oceanographers or some scientists, who knows, I don't know, maybe they're not. So it's good to just keep this in our minds and in our work, and not gloss over the uncertainties. The uncertainties are necessary so that the data can be useful to stakeholders, and the uncertainties complete the data. So they aren't just like this annoying side effort. They are the data, and they're crucial. And then just like a few quick sort of other little applications and features of data collection and PCC data. So one thing that's becoming more popular is real-time or near real-time use of in situ data, specifically optics in this example, while also environmental data, to look at features. So this is from a cruise last year in the South Atlantic, and we used the chlorophyll extracted from the ACS measurements and the IFCB images pretty much in real time to find the center of this eddy that we knew existed from satellite data, but it wasn't the exact location that we had because it had moved and the ship had moved and it was cloudy, etc. But we were able to kind of find the by processing the data using the inline analysis that Guillaume and Niels have worked on the ship and find this center of this feature. And there's more and more data that's available, that's in situ data that's available and valuable for algorithm development. So I'm sure you've heard of sea bass, there's Pangea, there's a variety, but this is one just from a researcher at the University of Washington. They collect picoplankton data using a continuous flow cytometer that they developed. It's their own instrument, but they have a really nice set of software, data repository, and it's well organized and useful for algorithm development if you're interested in the cells smaller than 5 microns, which are very important in the open ocean, even though they're not as interesting to look at as the diatoms. And then also this is a project that came out of that same group at the University of Washington, the Armbruster Lab, but it's a bigger, more collaborative project now, and where they're trying to collect data on phytoplankton microbes. So if you haven't heard of it, it's useful. And then a recently published data set for coastal and inland data, if you're interested in that, which is this Gloria data set. And it's all hyperspectral plus water quality parameters from all over the world. Lots of data and associated uncertainties. So just a couple of kind of conclusion slides to leave you with. How can we navigate all the challenges that come with trying to estimate PCC with the opportunities? And I think sometimes there's this tendency or for the community to try to say like, well, can we just decide on what the 8 phytoplankton groups are, or can we, how we need to put our stick in the sand and say this is what PCC is, and so that we can go forward. But I would just, rather than that, I would encourage you to think about what are you after, and it's perfectly fine to have some PCC algorithm that's tuned to your toxic dinoflagellate in your bay to support something in your community, and a global application that's going to look at a climate. It's going to be useful if in a climate model is just not useful locally, and vice versa. So just keep thinking about spatial and temporal variability and resolution and the question you're after. This is the message to myself too. Turns out if you try to do everything at once, it just doesn't work. So pick something and then use the data and the approach that's appropriate for that question. And remember the uncertainties. Okay, last 2 slides here. Major challenges. Oh, but they're going to go one at a time. Here they are. So as I mentioned earlier, the sensitivity of the methods, to uncertainties. And then as I mentioned earlier, the fact that the groups that we're after are often defined by proxy. So for example, phytoplankton pigments used to define PCC, that creates a ceiling on how accurate the PCC algorithm can be if you're really after the biomass of a group. Sufficient data sets, but more and more we have autonomous platforms and continuous instruments deployed on ships. And then thinking about what users actually need versus what we're measuring, and in the funding environment, there's, I would say, in my experience, increasing emphasis on the stakeholders and the end users, and basic research we always need for the sake of it, but we also need to think about what do people need and why, and how can we as scientists contribute to that effort and be useful in our short lives. And exciting opportunities. We'll just end on a high note. Lots of advancements in data collection. The instruments that you all learned about the last 4 weeks and the methods are totally different from when I took the optics class. Well, not totally, but there are lots of new approaches, and that's very exciting. And hand in hand with that is the increased collaboration with data scientists, software engineers. Like we need these tools so that we don't have a bottleneck with big data rolling in and limitation with even being able to do anything with it. So as Emmanuel has said, in recent years, we've become data inundated versus data limited in oceanography. And that really means something for how we approach the science and who we collaborate with. Hyperspectral satellite remote sensing, also UAV data. I didn't talk about here, but Patrick has a lot of experience using drones. I don't know if you did any of this class, but ask him if you have questions about using drones to look at ocean color. Increase in machine learning. Machine learning is not new, but the application of it in oceanography is becoming more popular, also obviously with increased computing power. Ancillary data, please think about, not working in a vacuum. This is also something I've picked up from Emmanuel, but we know something about the ocean. It's a beautiful color, but we know other things about what we expect to see there in the environment, in the physical environment, and the movement of the water. I mean, there's endless ways we could be combining all that data, and biophysical oceanography is becoming more popular, and it's, well, I don't know, but is a useful avenue to like link together different types of data. And of course, improved models data collection that allows to look at finer scales. So continuous measurements where we can look at detailed information across fronts, and more complex models that help us think about predictions of what we might expect, or what we might see in the future in the ocean. So in the spirit of Kurt Mobley ending with a picture or two, here's a picture from 2011. Meg, my TA, this is the lobster thing, so beer's appropriate. Many of these people are still my close friends and colleagues. They work at NASA, NOAA, they're professors, research scientists. I hope you all have had an experience these last few weeks where you feel like you want to stay in touch with each other, because it totally was life-changing for me to take the optics class, and I feel very fortunate. So here's Colin and Nathan Briggs. And here's Jeremy's kids who are now in college. Jeremy's somewhere in the back, maybe. Maybe Emmanuel's taking the picture. Yeah, good times. Oops, and thank you.